Hello dear students, we are going to discuss today on sources of impurities and their control. A compound is said to be impure if it is having foreign matter. Pure chemical compounds refer to the substances which is having no foreign matter, that is impurities. Purification of chemicals is an expensive process. Substances should not be purified more than required as it brings about waste of time, material and money. Pure is a relative term and Indian Pharmacopoeia and British Pharmacopoeia has been prescribed test for purity. The pharmaceutical preparation should be free from toxic and other impurities. Pharmacopoeia prescribe limit for harmful compound present in substances. The substances that are used in pharmaceutical field must be pure so that they can be used safely. But it is very difficult to obtain an almost pure substances. We find different substances and chemicals with varying degree of purity. So, today we are going to discuss different sources of impurities, their control and some of the effect of impurities on pharmaceuticals. Some effect of impurities are as follows. The impurities which are present in the substances may have the following adverse effects. Impurities may bring about incompatibility with other substances. Impurities may lower the shelf life of the substances. Impurities may cause difficulties during formulation and use of the substances. Sometimes impurities changes the physical and chemical properties of the substances. Therapeutic effect can be decreased, shows toxic effects after a certain period, injurious when present above certain limits. It may be change odor, color, taste of the substance. A list of impurities which are likely to be present in a given pharmaceutical substances can be easily compiled from the knowledge of the raw materials employed, the manufacturing processes and stability of the final product. Impurities may also arise from the physical contamination and improper storage conditions. The various sources of impurities in pharmaceutical substances are as follows. Raw material employed in the manufacturing. Second, method or process used in manufacture. Third, reagent employed in manufacturing processes. Fourth, use of reaction vessels. Fifth, crystallization related impurities. Sixth, stereochemistry related impurities. Seventh, synthetic intermediates and bioproducts. Eighth, formulation related impurities. Ninth, impurities arising during storage. 10th, mutual interaction among ingredients. 11th, atmospheric contamination during the manufacturing process. 12th, manufacturing hazards. 13th, instability of the product. Now, we will go one by one to each step. Now, first, raw materials employed in manufacturing process of pharmaceutical substances. Pharmaceutical substances are either isolated from natural sources or synthesized from chemical starting materials. It is essential to verify the identity of the source materials and to establish its quality. Otherwise, impurities associated with the raw material may be carried through the manufacturing process to contaminate the final product. In nature, minerals rarely occurs in a reasonably pure form. That is, aluminum ores are usually accompanied by alkali and alkaline earth compounds. Barium and magnesium impurities are found in calcium minerals and having lots of examples. Rock salts used for the preparation of sodium chloride is contaminated with a small amount of calcium and magnesium chloride. So that sodium chloride prepared from rock salt will definitely contains traces of calcium and magnesium compounds. Second, method or the process used in the manufacture. The process or method of manufacture may introduce new impurities into the final product arising due to contamination by reagents, catalyst and solvent employed at various stages of manufacturing processes. The new impurities may also arise from the reaction vessel and reaction intermediates. A known impurity 1,2,6-dichlorophenyl indolene 2,1 is formed in the production of a parenteral dosage form of diclofenac sodium if it is terminally sterilized by autoclave. The condition of the autoclave method enforce the intermolecular cycling reaction of diclofenac sodium forming an indolinone derivative and sodium hydroxide. The formation of this impurity has been found to depend on initial pH of the formulation. Third, reagent employed in manufacturing processes. Calcium carbonate contains soluble alkali as impurity which arises from the sodium carbonate employed in the process. Calcium carbonate is prepared by interaction of a soluble calcium salt with a soluble carbonate. Therefore, the final product calcium carbonate is liable to contain small amount of soluble alkali as impurities which were not removed by the washing process as shown in the given reaction. Calcium chloride when react with sodium carbonate forms calcium carbonate and sodium chloride in which the calcium carbonate is in precipitated form. Now, third, 3.1 is the solvents. Most of the pharmaceutical substances are prepared in solvated crystalline form. Small amount of solvents employed in preparation and purification of reaction intermediates or the final product may also result in contamination of the pharmaceutical substances. Water is the cheapest solvent available and is used quite frequently in preparation of inorganic pharmaceuticals. Water can be the major source of impurities as the different types of water containing different types of amount of impurities are available. 3.2 Residual solvent. Residual solvents are organic volatile chemicals used during the manufacturing process or generated during the production. Some solvents that are known to cause toxicity should be avoided in the production of bulk drugs. Depending on the possible risk to human health, residual solvents are divided into three classes. Class 1 includes benzene having 2 ppm limit, carbon tetrachloride having 4 ppm limit, methylene chloride having 600 ppm limit, and methanol is having 3000 ppm limit, pyridine with 200 ppm, toluene with 890 ppm should be avoided. Class 2 having N,N-dimethyl formamide with 880 ppm limit, acetonitrile with 410 ppm. Class 3 includes acetic acid, ethanol, acetone has permitted daily exposure of 50 mg or less per day as per the ICH guidelines. Fourth, use of reaction vessels. The reaction vessels employed in manufacturing process may be metallic such as copper, iron, cast iron, galvanized iron, silver, aluminium, nickel, zinc and lead. Glass and silica are also used in the construction of the chemical plants, but these days many of these are replaced by stainless steel and variety of other alloys. Some solvents and reagents employed in the process may react with the metals of reaction vessels, leading to their corrosion and passing traces of metal impurities into the solution, contaminating the final product. Fifth is crystallization related impurities. Based on the realization that the nature of structure adopted by a given compound upon crystallization gives a profound effect on the solid state properties of the system. The pharmaceutical industry is required to take a strong interest in polymorphism and solvatomorphism as per the regulations laid down by the regulatory authorities. The crystallization related impurities are always arising in finished products. Sixth is stereochemistry related impurities. It is important to look for stereochemistry related compounds, that is, those compounds that have similar chemical structure but different spatial orientation. These compounds can be considered as impurities in the APIs. Chiral molecules are frequently called enantiomers. The single enantiomeric form of chiral drug is now considered as an improved chemical entity that may offer a better pharmacological profile and an increased therapeutic index with a more favorable adverse reaction profile. The prominent single isomer drug which are being marketed includes levofloxacin having S-ofloxacin and levobuterol, R-albuterol and esomeprazole with S-omeprazole. Next is 7th, synthetic intermediates and bioproducts. Sometimes an intermediate substances produced during the manufacturing process may contaminate the final product. That is, sodium bromide is prepared by reaction of sodium hydroxide and bromine in slight excess. Here is the reaction: sodium hydroxide react with bromine and form sodium hydrobromide. The sodium bromate, an intermediate product, is reduced to sodium bromide by heating the residue with charcoal, as shown in the reaction. A sodium bromide react with charcoal and gives you sodium bromide. If sodium bromate is not completely converted to the sodium bromide, then it is likely to be present as an impurity. Next one is formulation related impurities. Many impurities in a drug product can originate from excipients used to formulate a drug substance. In addition, a drug substance is subjected to a variety of conditions in the process of formulation causes degradation. If the source is from an excipient, variability from lot to lot may make a marginal product unacceptable for reliability. Solutions and suspensions are inherently prone to degradation due to hydrolysis. In general, liquid dosage forms are susceptible to both degradation and microbiological contamination. In this regard, water content, pH of the solution, suspension, compatibility of anions and cations, mutual interactions of ingredients, and the primary containers are critical factors. Microbiological growth resulting from the growth of bacteria, fungi, and yeast in a humid and warm environment may result in the unsuitability of an oral liquid product for safe human consumption. Next is impurities arising during storage. A number of impurities can originate during storage or shipment of drug products. It is essential to carry out stability studies to predict, evaluate, and ensure drug product safety. Next one is mutual interaction amongst ingredients. Most vitamins are very liable, and on aging they create a problem of instability in a different dosage forms, especially in liquid dosage forms. Degradation of vitamins does not give toxic impurities. However, potency of active ingredients drops below pharmacological specifications. Because of mutual interaction, the presence of nicotinamide in a formulation containing 4 vitamins, mainly nicotinamide, pyridoxine, riboflavin, and thiamine, can cause the degradation of thiamine to a substandard level with a 1-year shelf life of vitamin B complex injections. As seen earlier, impurities in drug products can come from the drug or from excipients, or can be brought into the system through an in-process step by contact with the packaging material. For more drugs, the reactive species consist of water that can hydrolyze some drugs or affect the dosage form performance, small electrophiles like aldehydes and carboxylic acid derivatives, peroxides can oxidize some drugs, metals can catalyze oxidation of drugs and the degradation pathways. Last is leachable or extractable material can come from glass, rubber, stoppers, and plastic packaging materials. Next, atmospheric contamination during the manufacturing process. Atmosphere may contain dust, aluminum oxide, sulfur, silica, soot, etc., and some gases like carbon dioxide, sulfur dioxide, arsine, and hydrogen sulfide. These may contaminate the final product during the manufacturing processes. Some substances which are susceptible to action by atmospheric carbon dioxide and water may get contaminated with them during their preparation. That is, sodium hydroxide readily absorbs atmospheric carbon dioxide when exposed to atmosphere. We know that a sodium hydroxide when react with carbon dioxide, it forms sodium carbonate and water. Calcium hydroxide solution can absorb carbon dioxide from the atmosphere to form calcium carbonate. As shown in the reaction, So we can prevent the hydrolysis of sodium hydroxide by using calcium hydroxide solution. Next is manufacturing hazards. If the manufacturer is able to control and check impurities from the all above mentioned sources, there exist certain manufacturing hazards which can lead to product contamination. The various manufacturing hazards can lead to contamination from the particulate matter. The unwanted particulate matter can arising by a number of ways, such as accidental inclusion of dirt or glass, porcelain, plastic or metallic fragments from sieves, granulating, tableting filling machines and the product containers. The particulate contamination mainly arising from the wear and tear of the equipments. Cross contamination of the product. This manufacturing hazard has to be considered in the preparation of solid dosage form. Cross contamination of product can occur by airborne dust arising out of handling of powders, granules and tablets in bulk. Cross contamination is dangerous, particularly in case of steroidal and other synthetic hormones. Contamination by microbes. Many products like liquid preparations and creams intended for our topical application are liable to contamination by microbes from the atmospheric during manufacturing. For all products intended for parenteral administration and ophthalmic preparations, sterility testing is done and it provides an adequate control for microbial contaminations in such preparations. Microbial contamination can be controlled by adding suitable antimicrobial and antifungal agents. Errors in packaging. Similar looking products such as tablets of the same size, shape and color packed in a similar containers can result in mislabeling of either of both of the products. Instability of the product. Chemical instability. Impurities can also arise during storage because of chemical instability of the pharmaceutical substances. Many pharmaceutically important substances undergone chemical decomposition when storage conditions are inadequate. This chemical decomposition is often catalyzed by light, traces of acid or alkali, traces of metallic impurities, air oxidation, carbon dioxide and water vapors. All such decompositions can be minimized or avoided by using proper storage procedures and conditions. The photosensitive substances should be protected from light by storing them in darkened glass or metal containers, thereby inhibiting photochemical decomposition reactions. Changes in physical properties. Pharmaceuticals may undergo changes in physical properties during storage. There can be changes in crystal size and shape, sedimentation, agglomeration and caking of the suspended particles. These physical changes are not always avoidable and may result in significant changes in the physical appearance of pharmaceuticals and therapeutic effect of the product. Similarly, increase in the globule size of the injectable emulsions on storage may lead to fat embolism. Reaction with container material. The possibility of reaction between the container material and the contents cannot be ruled out as it constitutes a safety hazards. Preparations susceptible to reaction with metal surfaces, that is salicylic acid ointments, must not be packed in metal tube. Solution of substances which are alkali sensitive, that is atropine sulfate injections, must be packed in glass ampoules which comply with the test of hydrolytic resistance. Therefore, such preparations must not be packed in containers made from soda glasses. Now, temperature. The rate of chemical decomposition and physical changes of stored products depends upon the temperature. The susceptible substances may have temperature storage requirements assigned to them in order to protect them against undesirable decomposition. Now, the module 2, the control of impurities. Pharmacopoeial methods. Official monographs for pharmaceutical substances provide description and information in addition to prescribing standards for products and its storage conditions. An official monograph for a pharmaceutical substances generally included the following: title, chemical formula, chemical names, category, dose, description, solubility, storage. We can describe storage. It contains information regarding the storage condition of the pharmaceutical substances so that they can be guarded against possible contamination and deterioration. The precautions need to be taken regarding the effect of atmospheric moisture, heat and light are also indicated where appropriate in the individual monograph. Next, standards as determined by the assay. It specifies the quantitative purity of the official compounds. If an article does not comply with all the stated requirements, it is not of pharmacopoeial quality. Next is assay. It describes the official method for the quantitative determination of the active ingredients of the pharmaceutical substances and its preparation. Next is identification test. The purpose of identification test is to ensure the correct labeling of the substances. Identification tests are specific, but they are not necessarily sufficient in establishing the absolute proof of identity of the substances. If an articles taken from a labeled container do not meet to the requirements of a prescribed identification test, indicates that the article is either mislabeled or substituted. In some same monographs, more than one identification tests are given. Identification tests are generally based upon the combination of simple chemical test and measurement of the appropriate physical constants. There is a considerable overlap between identification tests and the limit tests. Limit tests are designed to ensure that the undesirable impurities are within the prescribed limits. Practically, a single identification test may contribute to identification as well as standardization of the substances. Chemical tests used for identification are basically quantitative, confirming to the presence of the substances under investigation. Physical constants such as melting point, boiling point, solubility, weight per ml, refractive index, optical rotation, viscosity, etc. have characteristic value for a given substances. They can be used in identification, checking, quality and maintaining standards of purity. Next is test for purity. Tests for purity for substances have been prescribed by the pharmacopoeias of the various countries in order to ensure reasonable freedom from the undesirable impurities. The so-called tests for purity are in fact the test for the presence of impurities in the substances and fix the limits of tolerance for these undesirable impurities. Tests for purity are not framed to guard against all the possible impurities, rather they provide appropriate limitations of the potential impurities only. Arsenic and lead are quite dangerous even in trace amounts, therefore very small limits of tolerance have been fixed for their presence in all pharmaceutical substances. Another factor in practicability of the commercial method of production of the substance meeting the requirement of a particular standards of purity. There are cases in which the limit fixed in the pharmacopoeia were later relaxed because they were found to be too difficult to attain by the available method of manufacturing. Next is assay. An assay method should be specific for the substance or chemical species being examined. Assay tolerance play an important role in fixing standards for pharmaceutical substances. They include the limit of error of the actual assay process, the limit of tolerance of manufacturing process for the particular dosage form, and the sampling errors. The limits of errors of the assay process depends upon the method employed for manufacturing. Next is limit tests. Limit tests are quantitative or semi-quantitative tests designed to identify and control small amount of impurities which are likely to be present in the substances. They involve simple comparison of opalescence, turbidity or color produced in test with that of fixed standards. Sources of control of impurities. Washing. When water soluble substances have to be washed away and water insoluble substances is needed. Example is chalk from native calcium carbonate. By drying. Generally dried in air anhydrous chemicals, vacuum drying has been an impurified unit operation and need to care and precautions so that chemicals may not deteriorate due to oxidation, caking or mold growths. Recrystallization. Recrystallization is the most common method of purifying soluble salts. Solution is allowed to cool slowly, after which crystals of greater purity could be obtained. Sublimation is a process applicable to very few substances, for example arsenic trioxide, iodine and mercury chloride. Sublimed sulfur. The organic compounds purified by this process are camphor and benzoic acid. Module 4. Conclusion. The ultimate objective is that the pharmaceutical substances completely free from the undesirable or toxic impurities should be of reasonably good purity, ensuring therapeutic safety. There is an ever increasing interest in impurities present in APIs recently. Not only purity profile but also impurity profiling has become essential as per various regulatory requirements. The presence of the unwanted chemicals, even in a small amount, may influence the efficacy and safety of the pharmaceutical products. In the pharmaceutical world, an impurity is considered as any other organic material besides the drug substances or ingredients arise out of synthesis or unwanted chemicals that remains with APIs. The impurities may be developed either during formulation or upon aging of both APIs and formulated APIs in medicines and can be explained using multidisciplinary approach. The ICH has also published guidelines for validation of method for analyzing impurities in new drug substances, products, residual solvents and microbiological impurities for the final products to be used by human being. Hope you all enjoyed this informative lecture. Thanks for your attention. Keep studying and goodbye students.